wabarakatuh. Good afternoon, everyone. We are from Group B. We will present our laboratory activity report about PDA mediation and APH isolation from plant tissue. And before we start our presentation today, I will introduce our member. First is Johanes Matanari, student number 005. Me, Farika, Arretama, Pujiarini, student number 060. Amalsya Ikarohmi, student number 077. Sara, Vanesya, student number 131. Gracela, Nanda, student number 169. Rustiana, student number 192. Serina, student number 198. Desty number 233. And Isna, Yuni, Istierin, student number 242. First of all, Farika, member of the cashew family and one of the most important and widely cultivated fruits of the tropical world. The process of cultivating mango plants cannot always be separate from disease and pests. To controlling the disease of mango plants, except using synthetic insecticide, we can also using biological agents like Trichoderma sp. Biological agent is one of the control solutions for disease in mango plants.
Use of Trichoderma to control plant disease is not harmful to the environment unlike chemical pesticides. Trichoderma strains have also been recognized for their ability to increase root growth and development, crop productivity, resistance to Therefore, it is important to investigate the diversity of Trichoderma, since such information can lead to the isolation of Trichoderma species having higher antagonistic efficiency and development of better biological control methods to manage plant pathogenic fungi. Next. Mango leaves consist of two parts, namely the petiole and the leaf body. The leaf body has buds and veins between the leaf buds, and the veins are covered with leaflets. Mango leaves are covered by a thin skin that isn't easily visible to the naked eye, called the epidermis, in which the leaf mouth of Arostomata is located. Overall leaf length between 8.47 until 23.82 centimeter, leaf width between 3.22 until 6.04 centimeter, leaf area between 30.20 until 101.10 centimeter square.
The next one is Trichoderma fungus morphology. Trichoderma sp is an antagonist fungus that has the ability to inhibit the growth of pathogenic fungi. The antagonist mechanism of the fungus Trichoderma sp against soil pathogens can be in three ways, namely producing extracellular enzyme beta-1 1.3 gluconase and chitinase, producing trichodermin toxin and producing glycotoxin and pyridine antibiotics. Next. Trichoderma sp has a dark green color with the color on the PDA media. The colony shape of the isolates spread irregularly and have uneven colony edges. Microscopically, isolates of Trichoderma sp have septate hyphae, many and branches conidiospores, hyaline, semicircle conidia, and clustered between hyphae.
The next is PDA medium, potato, dextrose, agar. PDA medium is a solid medium consisting of three main such as potato, dextrose, and agar. Potatoes are used as a source of carbohydrates, vitamins, and energy. Dextrose as an additional source of energy. Agar used to make the medium solid. Based on the consistency, the media is divided into three. The first, solid medium, requires 12 until 15 grams of agar for 1000 ml of media, and then the second, semi-solid medium, media with only 50% or less of solidifying agent or agar than it should be. And the third, liquid medium, media in which no solidifying agent or agar is added. Next. PDA medium have advantages and disadvantages. And the advantages is this media is in instant form because this media is made by a certain factory or company which is already to be used. And the disadvantages is are expensive, hygroscopic, and can only be obtained in certain place.
And then there are the tools and materials that we need to make the PDA materials. The PDA media materials is our 200 grams of potatoes, aquades, water, 20 grams of dextrose, 15 to 20 grams of gelatin, and also aluminum. And then the PDA media creation tools are knives, Erlenmeyer, rubber bands, and autoclave. And the APH isolation materials, we need mingle leaf tissue, water, alcohol 96%, aquades, tissue, medium PDA plus lactic acid. And in the APH isolation tools, we need cutter, petridis, tweezers, spiritus, and also laminar airflow. Next. So this is how to we create the PDA media creation. The first one is by washing 20 grams of potatoes and peel the potato. After the potato peeled, we cut the potato into dice and then boiled using 1 liter aquades until the potato is soft. And then the boiled water from the potato is transferred to the Erlenmeyer 2 liter. After that, we can add 20 grams of dextrose and 50 to 20 grams of agar-agar to the Erlenmeyer after the potato stew water is warm. Next. And the next step is after the solution filled Erlenmeyer is heated by dipping it in boiling water while stirring until the processor and gelatin are dissolved. We can take 200 milliliter of the solution, then transfer it at the Erlenmeyer 215 milliliters. After that, we can close the Erlenmeyer using aluminum foil, and we can add the pepper and also rubber bands. After that, we can sterilize the PDA media in that Erlenmeyer using the autoclave at 121 degrees Celsius for 20 minutes, and the media is all ready to use. Next.
And then step 4, isolation of trichoderma from mango plant tissue. Step 1 is the material to be isolated is cleaned of dirt using running water, and then cut the healthy part of the leaf into small pieces, and then clean the dirt on the surface of the leaves. Small pieces of leaves put in the petri dish that has been poured with 99% alcohol and soak for 1 minute. After that, rinse it using aquades for 1 minute and line it using tissue. Next. Furthermore, the pieces of leaves are transferred into a petri dish containing medium PDA plus lactic acid, and then the leaf pieces were incubated at room temperature for 2 until 4 days. And then colonies of growing fungi are observed and transferred to new media. And the last step, fungal colonies were incubated at room temperature until pure culture were obtained. Next.
Okay, this is the observations research. The picture in left one is leaf isolate on 20 March, and the right one is leaf isolate on day 4. bar, and the last one, the research of purifications of leaf isolate on 29 March, and the last picture is the last observations research. Next. Okay, this is our discussions. 4 days as after incubations, there is a white antagonist fungus that grows around isolate. The fungus is indicate not Trichoderma sp fungus. The fungus is purified by replacing the new PDA plus lactic acid media, and purifications incubations research indicate the absence of Trichoderma sp fungus characteristics on the media, failure of Trichoderma isolations, possibly due to the isolations process is not done sterile, or on the mango leaves themselves are not contained Trichoderma sp fungus. The mango leaves have a slippery surface, so fungus can colonize. And the last, the failure in the isolation of plant tissue can also occur because at the time of the purifications, the tools used are the less sterile, so that contaminations can occur easily. Next.
So from our discussion, we can conclude that Trichoderma sp is an antagonistic fungus. This fungus has significant antagonistic potential to a wide variety of pathogenic fungi. PDA media is a solid medium consisting of three main ingredients consisting of synthetic and natural materials, namely potatoes, dextrose, and agar. Based on the observation, on 4 days after incubation, there is a white euthanized fungus that grows around isolates. The fungus is indicate not Trichoderma sp fungus. Failure of Trichoderma isolation probably caused by the isolation process is not done sterilely, nor microorganism on mango leaves itself is not contained Trichoderma sp fungus. Other causes can also be caused because the tools used are less sterile so that contamination is easy to occur. Next. Thank you for your great attention. Thank you for listening. May all those we have shared be beneficial for all of us. Thank you and wassalamualaikum warahmatullahi wabarakatuh.